A very good morning to all of you. I know that it's not our regular lecture slot, but we had to make some adjustments to adjust part one lectures. That's why just for today, I'm over here at 11 a.m. and my lecture will be engaged by Shweta ma'am. We have just uh, interchanged the slots for today. So I know that we are supposed to be discussing PCR for diagnostics of genetic disorder and karyotyping. But to be frank enough, I am looking in for real good material for karyotyping as yet, because I want that section to be a little more uh, elaborate and you can say interesting, because I feel that it is something very, very significant. So maybe I will take some more time for that to come up with a good content. And till then, what we will do is we will start with our discussion for gene therapy. Obviously, gene therapy is something which is very crucial, very interesting, an upcoming area since a very long period of time, actually, because it's not a new concept. It's a concept which has been in existence for quite some time now. But the success stories are really very less. And that in turn makes us question whether really gene therapy is going to turn out to be the hope of the future, or are we going in the wrong direction? But there are stories strong enough to support the potential that gene therapy basically possesses to cure several genetic disorders. And when I say genetic disorders, it's not just the regular genetic disorders that we have heard of, but also the rarest of the rare genetic disorders. People have been making use of gene therapy as a tool to not just treat conditions like diabetes, which might be genetically predisposed, wherein individuals are born with diabetes. Let me tell you that that's what we basically refer to as juvenile diabetes, wherein the child is born with diabetes because they are genetically predisposed in such a way that the insulin that they produce is either defective, because of which it doesn't interact with the receptor, or the insulin is not produced at all. And let me tell you, they have a tough life. I don't know how many of you know about this, that they need to take insulin shots throughout their life because their body is not going to synthesize insulin, right? And in several cases, artificial pancreas are, spoken about as a tool to deal with this particular condition in people who cannot synthesize insulin at all. Now, let me tell you, you will see such individuals with insulin pouches on their body, wherein it's like a normal pouch with insulin in it, and there is a catheter which is inserted through the abdominal area so that the insulin is delivered from time to time. So it's a pump which delivers a little amount of insulin, periodically into the system so that they have a constant supply of insulin within them. Now, this is something that we all need to understand, that under such circumstances, we think, okay, gene therapy is going to be so crucial for such individuals because if it is genetically predisposed and if we are able to give them the ability to produce insulin on their own, it's going to be great. But let me tell you that gene therapy has come as a hope, not just for this, but also for cancers which are genetically predisposed, because we know that there are types of cancers which are inheritable. And under those circumstances, when we know that a gene is responsible for making me susceptible to cancer, if gene therapy can help me cure that specific condition, I think there is nothing like that. It's going to be really great. Similarly, if a person is blind, has lost, or does not have vision right from birth. And that is basically because of genetic predisposition, because of a defective gene. Imagine if we are able to restore vision in such people using gene therapy as a tool. That's going to be phenomenal, right? Similarly, there are so many people who are born with several such disabilities, such conditions because of genetic defects. So if I am able to revert that using gene therapy, obviously it's something which is going to be a ray of hope for such people. But unfortunately, the rate of success is not that great, because of which people have always been very much doubtful about whether to go for gene therapy or not. And secondly, it's mostly a trial and error method, because of which the overall expense of the technique is going to be extremely high. It's going to be phenomenally high. And moreover, if something like this is very expensive, affording such a technology in a developing nation becomes even more difficult. So in this section, we will be learning about how gene therapy is done, what are the approaches, which vectors are made use of to deliver a gene in an individual, what are ethical issues related to gene therapy, what are limitations that are seen with respect to gene therapy, and what currently is happening out there. So that's exactly what we will be discussing in this specific section. So without any further delay, I'm sharing my screen with you guys, and we will start with our discussion on gene therapy. So there are almost 4,000 known genetic disorders. The number obviously may be a little different from what we are saying right now, but 4,000 by itself is a huge number. And whenever we speak about genetic disorders, the basic problem is that there is no known therapy which is going to solve or cure this situation completely. That's the biggest problem, right? There's no effective therapy out there. And even when I talk about conditions like diabetes, I don't know how many of you know this, and we all know this fact that the number of diabetic patients in our country, or rather in the entire globe itself, is enormously huge. I'm sure ये बात आप सबको पता है, और इसका एक ही इलाज है, वो है इंसुलिन प्रोवाइड करना, इंसुलिन एडमिनिस्टर करना. लेकिन हम में से कितने लोग ये चीज जानते हैं कि इंसुलिन जो इंजेक्ट किया जाता है इंट्रावेनसली, आपने देखा होगा कि डायबिटिक पेशेंट्स जो होते हैं उनके लेवल ऑफ डायबिटीज के हिसाब से आइदर उनको ओरल एडमिनिस्ट्रेशन ऑफ इंसुलिन दिया जाता है या इंट्रावेनस एडमिनिस्ट्रेशन ऑफ इंसुलिन दिया जाता है. So normally जिनका level of sugar level in the blood is very high, they are told to take the intravenous shots because the oral administration may not be really very effective. So they are told, they are advised to take the shots before every meal that they have. Okay. Now, I don't know if we have discussed this out earlier, but let me tell you that you are injecting pure protein into your system. Okay, you're injecting a pure protein into your system probably thrice in the day, considering breakfast, lunch, and dinner. Okay, and we need to understand that for a longer period of time, if we are continuing this, if we are continuing administering insulin, it is not good for my kidneys. Because please understand that breaking down proteins in our system is a comparatively slow process. Our system is not built to eliminate out proteins effectively from the system because our system is built to utilize amino acids which are present in the protein that we take up. okay, and use those amino acids during the process of transcription and translation, that is during protein synthesis. But when I'm administering pure proteins directly into my blood, it's going to increase the load on the kidneys. And let me tell you that it's a known fact that people who take insulin for 15, 20 years, the kidney starts malfunctioning. It's a known fact. Even people who take statins, statins are the drugs which are prescribed for blood pressure. It's known to have an effect on kidneys. And if you talk to your physicians, and if you ask them that is this true? Is it true that insulin being administered intravenously or statins being taken, for patients who have high blood pressure issues and they have to take this medication from time to time, is it going to harm me in the long run? Is it true that it's going to affect my kidneys? The answer is going to be yes. So if you ask them the next question, that when you know that it's going to affect my kidneys, why do you give it to me? You know what will be the answer that you will get from the physician? This I'm talking from my personal experience. And I came to know about this first, and when I spoke to my family physician, and I asked him that, you agree that this is going to harm an individual. Why do you prescribe it? So his answer was that if I don't prescribe it right now, he or she is going to die sooner. So it's better that at least we are extending their lifespan. So ultimately, treating ourselves for one disorder is pushing ourselves to the other. And that is disastrous. So we need to look for safer alternatives. We need to look for alternatives, especially when it is congenital, when it is something which is related to my genetic predisposition. I need to look at how could I actually treat it. Like a lot of people are predisposed to high blood pressure as well, obesity as well, because it runs in the family. So no drugs is going to help me. I have to control my metabolism. I have to work on my metabolism to alleviate any kind of consequences that I would face because of my genetic predisposition. Now, when you talk about defective genes, you need to understand that any individual needs to have at least one good copy of the gene, because obviously, because we say that we all are diploid, we have two copies of all the genes, right? Because we have two copies of every chromosome. So one good copy of the gene is enough to not make me suffer from that particular disorder. So for me to be suffering from that condition, I know that I need both the defective copies. Only then I'm going to be referred to as diseased. But in some cases, we need to understand that if the disease is coded for by a dominant allele, then even one copy of it is enough for causing that condition in me. But most of the genetic disorders, we know that it's caused by the recessive allele. So I would need both the copies of the recessive allele for me to be diseased. If the disease is encoded by a dominant allele, then I am gone. Even one copy of that defective dominant allele is enough for me to be diseased. Now, if the defective gene runs in the family, according to Mendelian genetics, you know that there are, if it's coded by the recessive allele, then there is this 25% chance of offspring being diseased, right? But if it's encoded by a dominant allele, then the risk increases. You also know that if the disorder is related to genes of X chromosome, if it is X-linked, you know that males are more prone to it, right? Because males have a combination of XY. So they have only one copy of X. So it's obvious that if that gene is defective, then it's gone. They are going to be diseased. Like in case of color blindness, case of hemophilia, all these are X-linked conditions. So if the X chromosome received by the male offspring contains that defective gene, they are going to be suffering from that condition. But in case of females, because there are two X, they need to have the defective gene in both the chromosomes for them to be diseased. If they have one chromosome which carries the defective gene and the other is normal, they are going to be referred to as carriers. I'm sure you all know about this, and it's just a quick revision and brush up of the concepts that you have learned earlier. So let us move ahead. What are the current methods of treating genetic disorders? Obviously, that depends on what the condition is, what the disorder is. So it could be, if not complete cure, it could be treatment approaches to deal with the symptoms so that the patient doesn't have to suffer. So we are trying to lower down the intensity of the symptoms. So symptomatic treatment could be possible. In some cases, it could be a surgery-based treatment which would be necessary. In some cases, you may have to provide them specific metabolites because they cannot produce it. In some cases, if a genetic predisposition is making an individual to make toxic metabolites, then you will have to see how to flush those toxic metabolites out. If the genetic condition is damaging an organ, how to replace it. And also, if a specific gene is defective, can we actually restore the normal form of the gene? Or if not the gene product. So if we could administer the gene product, like in case of diabetes, you are providing insulin externally. But obviously all this is not a cure, right? And they have their own drawbacks and side effects. Because if you are administering something externally, what is the concentration? What should be the threshold concentration in them? All this would matter a lot. So all of these techniques would have their own drawbacks, and surely it's something that they will have to do lifelong, because it's not a cure ultimately. So they will have to carry all that treatment approaches throughout their life, and if that is expensive, it may not be really affordable for everyone, right? So the approach genetic engineering has brought in front of us, or gene therapy techniques has brought in front of us, is, can I really work or correct that genetic defect such that जड़ से हमने problem को खत्म कर दिया? Is that possible? So indeed it is possible. There is replacement therapy that is basically carried out right now. So these are all current practices before we go to the gene therapy aspects. So for hemophilia, you should be knowing what hemophilia is. Hemophilia is called as bleeder's disease. So the problem is with blood clotting. Their blood doesn't clot. Okay. So even if a safety pin pricks them, they are going to bleed continuously because their blood just doesn't clot. So the average clotting time in human beings is around 8 minutes. That's the average, okay? Some people it might be just in a minute or two. But in some people, it takes a lot of time for the blood to clot. So average time is 8 minutes. But these are those people, hemophiliacs are those people in whom the blood doesn't clot. So they could bleed to death. So they need to be provided with the coagulation factors so that if they have injury, these coagulation factors which have been administered externally, that will help them to carry out blood clotting so that they don't bleed to death. You would have also known of thalassemia, wherein there is a defect in production of hemoglobin. So unfortunately, these people have to be constantly on blood transfusion. You should be knowing about this, that these people need blood transfusion almost every 20 to 30 days. And आपने काफी बार पहले, मतलब अभी तो नहीं आता टेलीविजन पे, मैंने तो नहीं देखा है, बट पहले काफी बार ये एड आता था टेलीविजन पर, जहां पर एक लड़की हर एक को जाके थैंक यू बोलती थी. And the one person asks her that, why are you thanking me? So she says that I am thalassemic and I need blood transfusion, quite frequently. So I don't know who donates blood. So I thank everyone. and that was to encourage people to go for blood donation. So thalassemic people have a lot of consequences because of this constant blood transfusion. Jaundice is a very common symptom in them, constant jaundice, because they are getting administered a lot of blood in their system, and too much of RBCs are lysed in, and hemoglobin is broken down to biliverdin and bilirubin, too much of bile salt production, and that leads to jaundice. And there are several other side effects and consequences, and it's really very a sad condition, to be frank enough. There are also individuals who may have genetic disorders wherein their body doesn't produce immunoglobulins. Imagine, that's the worst situation. So that's called congenital hypogammaglobulinemia, which is deficiency of immunoglobulins, and they will have to be administered with immunoglobulins externally. Now, that's a big task. So their immune system is going to be pretty weak. Also dwarfism, wherein height doesn't grow. They don't grow in their height, and basically because of lack of growth hormone produced by pituitary gland. So they may have to be given shots of growth hormone. Other than that, we have already discussed about diabetes, so I'm not going to repeat that. Let's quickly go ahead. Thyroxine in case of hypothyroidism, wherein the thyroid doesn't function efficiently, right? Then you have something called as cortisone or 9-alpha fluorohydrocortisone. This is basically done when the adrenal gland doesn't work effectively. Okay, and there is adrenal insufficiency. So people will have to be administered cortisone. And this is basically very important a system for a basic reason that you all should be knowing what the function of adrenal gland is. Does anyone know here what's the function of adrenal gland? Their basic job is to regulate what? Is anyone writing something in the chat box? Let me check. Yeah, anyone? Adrenal gland. What does it regulate? Blood pressure. Yes. So indeed, Princey, it's located on the top of both the kidneys, and its basic job is to maintain osmotic balance, thereby controlling blood pressure. Also is very, very important for regulation of metabolism and enhancing immunity. So if adrenal gland doesn't function and it doesn't produce cortisol, then you have to provide cortisone. Also, you would have heard of severe combined immunodeficiency disorder, SCID, right? It's caused by ADA deficiency. What is ADA? Anyone? Adenosine deaminase. Adenosine deaminase is a very, very important enzyme for purine metabolism. Okay, and it's an enzyme which is found across kingdoms. It's found in bacteria, plants, invertebrates, vertebrates, mammals. And ADA is an extremely significant enzyme, because it converts deoxyadenosine, because whenever DNA is broken down, जब भी DNA को तोड़ा जाता है, whenever old DNA molecules are being metabolized, deoxyadenosine is produced, and deoxyadenosine is extremely toxic to lymphocytes. Okay, so adenosine deaminase is going to convert deoxyadenosine into a molecule called deoxyinosine, which is not at all toxic. Okay, but अगर ये ADA gene defective है और अगर ADA enzyme नहीं बन रहा, adenosine deaminase नहीं बन रहा, तो deoxyadenosine accumulate होने लगेगा, और अगर deoxyadenosine accumulate होने लगेगा तो वो मेरे immune system के cells को destroy करना चालू करेगा. So inka immune system is extremely weak, people who suffer from SCID. And you should be knowing that the very first gene therapy was tried out in this particular case of SCID only. You would have heard about this in your UG classes. And it turned out to be a failure, right? And that girl lost her life. So other than that, you would also see there are several metabolic conditions related to genetic defects, like phenylketonuria, tyrosinemia, wherein phenylalanine and tyrosine cannot be metabolized respectively. There is galactosemia, fructose intolerance, lactose intolerance, where galactose, fructose, and lactose cannot be metabolized respectively. There are also urea cycle defects wherein nitrogenous wastes accumulate, toxic nitrogenous wastes accumulate in the system. And all this could be really harmful to some of the vital organs of the system. So under such circumstances, dietary changes are suggested so that they avoid foodstuffs containing or which are rich in these components. And otherwise, they are given enzymes which they lack to metabolize these. Like in case of lactose intolerance, you all would be knowing that lactose cannot be digested by these people. So they could be provided with lactase enzyme, which is responsible for breaking down lactose effectively. So they could be provided with enzymes to deal with it, or they would be suggested to stay away from foodstuffs if they don't want to get administered with enzyme. In several genetic conditions which are related to blood, bone marrow transplantations would also be suggested. Okay, other than that, if there are conditions wherein vitamins are not being synthesized, then in those circumstances, those vitamins could be administered, like cobalamin, cyanocobalamin, riboflavin, and biotin. These are very important coenzymes. They function as coenzymes for several metabolic enzymes, and their absence could hinder the process of metabolism. So that could be administered. But obviously, all of this is going to be, something which is lifelong. It's not a cure. It's just to deal with the symptoms that are seen in this specific condition. So there will be a continuous treatment needed throughout the life, and that's going to increase the cost of the treatment, right? Now, when you talk about processes like blood transfusion, bone transplantation, we know that it involves other risks also. Like in case of blood transfusion, we know that always there is a scare of transmission of infectious diseases. Also, in case of transfusions, higher iron contents could occur in an individual after repeated transfusions, which is very dangerous again. There could be other side effects like jaundice, which we have already discussed. So what are the alternatives that we are looking for? First, with recombinant DNA technology coming into the picture, and our understanding of genetic disorders has in a way diversified because of the Human Genome Project, because we know that what are the normal genes and what kind of mutations could lead to a defective condition. So people have come up with gene therapy wherein they are thinking of introducing new set of genes into mammalian cells. So you're trying to introduce a functional gene, a healthy gene into the individual so that they could, produce the enzyme that they are lacking and the condition could be cured. But is it that easy is the question. So there are 4 potential approaches to gene therapy. Number 1 is that you add or you introduce a normal gene without replacing the mutant gene. So you're not replacing the mutant gene at all. The mutant gene will remain in the individual. What you are doing is that you're just introducing a normal copy of the same gene so that they will now be able to produce the gene product, the normal gene product, the functional gene product, and that would deal with the condition. Or you replace the mutant gene sequence by a normal gene sequence using recombination techniques. So in this scenario, you are getting rid of the mutated gene. You are not letting the mutated gene remain behind. You are replacing it with a normal copy. Okay. or you look for alternative pathways such that you don't have to do any gene therapy, but you are trying to induce an alternative process to produce a similar kind of a protein which would perform the same function, which is very difficult to do. Or you alter the regulation process of the mutant gene or the normal gene. If if the expression of the mutant gene is dangerous, and if you're trying to regulate it such that it doesn't express too much, and if the problem gets solved, that is also fine. So all of this, obviously, which approach you will choose will depend on the condition. Okay, and we will discuss that as we go ahead. So don't worry about that. So which approach you will make use of will depend on what condition you are dealing with. Now, what does that mean? So before that, have a look at this very important concept, wherein Indian government had sanctioned the very first gene therapy project, which was called the suicide gene therapy, which was against the oral cancer cells, wherein the gene therapy approach was to make the oral cancer cells commit suicide or go into apoptosis. And this was led by Rita Mohelkar, who is from ACTRAC. She has a very active lab in ACTRAC, and the first funded project, the government sanctioned project for gene therapy, started in the field of cancer. And what are the challenges? The first challenge is how to deliver the gene. And especially if it is to be delivered to a specific cell in the system, the problem increases. Because accessibility is a big challenge. How will I make the gene reach there? And more importantly, only there and not anywhere else. So I need a very effective vector for delivery of the gene. But at the same time, that vector has to be safe. I cannot compromise on the safety aspect. So no doubt, for specificity, such that the gene is delivered to a specific type of cell, viral vectors are the best choice. Because we know that viruses are highly specific towards their host cells. So you can make use of a specific virus which will deliver the gene at site. Retroviruses have been, the choice for specific gene therapy. But the problem with it is that retroviruses can act only on dividing cells, on dividing host cells. So if you want to deliver these cells to, or rather these genes to cells of the host which are not actively dividing, then retroviruses are a bad option. So for non-dividing cells, you will have to go for adenoviruses, because most of the body cells, they would be in the resting stage. Actively and constantly dividing cells in your body are, very specific and limited in number. Now, another thing that we have to consider as a challenge is, once the viruses carry the gene, the insertion could be very random on the DNA. So where exactly it gets inserted is a big question mark. So insertions could be random. Other than that, as you're using viruses, you should not forget that they have a chance to elicit an immune response. And if an immune response is elicited even before this virus could actually go and deliver your gene, then the entire process is going to turn out to be futile. Also, another vector that people have been thinking of and working on is cationic liposomes, which could fuse very easily with the cells and deliver the gene. But again, as it sounds, it's not that very simple to do it. Okay, and other than that, people have also been working with biolistic techniques. I think we have discussed about this earlier in the last semester also, the gene gun technique, wherein you are delivering the gene to a specific site using the biolistic method, right? So these are all different points about gene therapy, and now we are talking about the correct candidates for gene therapy. So first of all, जीन थेरेपी के लिए या कौन से कंडीशन में हमको जीन थेरेपी अपनाना चाहिए, this depends on what risk is involved in this. Okay, and the disorder is something which cannot be cured by any current strategies. For such conditions, only gene therapy should be opted for. You should also see to it that there are not too many complicating variables, because then if something goes wrong, the situation might turn out to be even more worse. Right? So you should opt for only those disorders where you see the potential of cure. Okay? Where you feel that, you know, this could be cured by doing a corrective therapy, or by replacing a gene, or by introducing an alternative copy of the healthy gene. So if you feel that it works in either recessive or dominant conditions, you could go for that. But yes, to do this, you should have the complete knowledge of the gene first. You should know the exact normal sequence of the gene first. And you should know that which mutations in this gene leads to what kind of a genetic disorder. That kind of data is now available because of Human Genome Project. And that data has to be handy, and that data has to be clear in your mind when you are opting for a gene therapy process. Now, in an individual, when you are carrying out, see, gene therapy is going to be tailor-made process. It's going to be a highly specific process for a patient. So you have to know the patient in and out. The defect in the gene has to be known in and out. It has to be well defined. Also, the tissue where you have to administer the gene, where you want to deliver the gene, that needs to be very much accessible. Okay? And those cells where you have delivered this gene, it needs to be self-renewable cells. It shouldn't be that आज मैंने जीन इंसर्ट कर दिया लेकिन कल वो सेल मर गया तो हो गया काम, वापस हमको जीन थेरेपी करना पड़ेगा. ऐसे नहीं होना चाहिए. Those cells should be renewable so that they pass on that gene which you have introduced to the next generation of cells also, so that you don't have to carry out this process time and again, because it's going to be expensive. And every time it may not turn out to be successful. So they should be done in self-renewable cells, right? And these corrected cells should show some selective advantage as compared to the other cells which were not corrected, because you cannot transfer, you cannot administer the correct gene to every cell. It's not possible, right? While if you are not trying to replace the defective gene, but you are letting the defective gene be, and you are trying to insert a normal copy of the gene such that now the cell has both a defective copy and a normal copy, you call such process as gene augmentation. Augment का मतलब ही होता है supplementary कुछ provide करना. तो already जो gene defective है वो रहेगा उनके अंदर, वैसे ही रहेगा. हम सिर्फ क्या करने की कोशिश कर रहे हैं? We are just trying to provide them a healthy copy. Now ये कब करना चाहिए? ये भी समझने की कोशिश करो. अगर डिफेक्टिव जीन की वजह से मुझे कुछ नुकसान है, अगर वो डिफेक्टिव जीन एक ऐसा प्रोडक्ट बनाता है जो मेरे लिए जहरीला है, जो मेरे लिए टॉक्सिक है, then my approach should not be augmentation. फिर पागलपंती होगा अगर मैं augmentation करने की कोशिश करूंगा. उस कंडीशन में मुझे उस डिफेक्टिव जीन को वहां से निकालना चाहिए, मुझे उसको रिप्लेस करना चाहिए, क्योंकि वो जो कुछ भी प्रोडक्ट बना रहा है वो मुझे हार्म कर रहा है. That's harming me. So augmentation will not work there. In that circumstances, I need to correct that situation. I need to get rid of that gene. But if that defective gene is not causing me any harm as such, it's just that it's not able to produce a functional protein because of which I'm suffering from some consequence, then I can let the defective gene be as it is. And I'm just somewhere else on the DNA inserting a normal copy of the gene. That's what I will refer to as augmentation process. Are you understanding this concept? All of you? Okay. So have a look at this. So gene augmentation by itself is gene replacement, but replacement in true sense doesn't mean that you are replacing the defective gene with the normal gene. It's just that you're inserting the normal gene somewhere else on the DNA while the defective gene is still there in place. So do not get confused by the term replacement. This is suitable for recessive disorders or defects wherein the one gene is defective. There would be no recombinational event to replace the defective gene. Okay, it's going to be a non-specific insertion that's going to be done. It may not be very much useful in case of dominant nature of the gene, because if the defect is because of a dominant allele, then getting that dominant allele out would be very important. Getting rid of it would be very important. Because वो अगर तुमने वैसे ही रहने दिया और साथ में एक नॉर्मल कॉपी दिया, लेकिन वो डोमिनेंट अलील अभी भी वहां पे तहलका मचाएगा, तो वो नहीं चाहिए मुझे. ऐसे कंडीशन में ऑगमेंटेशन इज नॉट गोइंग टू बी इफेक्टिव, राइट? सो obviously this is going to be working at the root level of the defect because you are trying to give them a normal copy of the gene. While in case of corrective gene therapy, it's actually getting rid of the defective gene. So you are inserting the normal gene at specific site. So it has to be an induced recombinational event. So your gene of interest would be flanked by regions which are complementary to the DNA strand, and where the gene has to be inserted, and you bring about a recombination. Very defective for gene disorders which may be dominant. Though extensive study is still on about how you will insert it to specific sites, but this question has been answered to a great extent with a lot of advances being done in genetic engineering and also CRISPR-Cas now. Okay. And in a way, you could get rid of the mutant gene in this process and get it removed and place the normal gene at the location. So that's the corrective gene therapy, or correcting the defect there. Now, obviously, whenever we talk of gene therapy, the question arises is there are two types of it: somatic therapy and germline therapy. So somatic therapy obviously is going to mean that the correction is going to be involved in the somatic cells. So the body cells basically. And if, you know, most of these gene therapies are going to be somatic therapies because we want the individual to, you know, be treated from this condition. So most of the gene therapies targeted towards somatic cells only, and it won't involve ethical issues until and unless you are, you know, in a way harming the individual in that process. So there is no ethical issue ideally involved. Though a lot of technical expertise would be needed for gene therapy to be performed effectively, but the changes are confined to the recipient only. It's not going to be passed on to the future generation because you're doing these changes in the somatic cells. Okay. And as I said, you cannot insert the correct gene in all the cells of the patient. That's not possible. It's going to be inserted into specific cells. So they are tissue specific. And that too in the tissue, I may not be inserting it in all the cells of the tissue. It may not be really feasible. Specific number of cells in the tissue, we are trying to insert the healthy gene such that the normal protein or the functional protein starts getting expressed. But in some situations, the gene therapy may be targeted on the germline cells. And the moment you do this, there are a lot of ethical issues that come into the picture. Okay, and there are going to be technical difficulties also to modify the germline cells. Now, ethical issues are basically because the changes are going to be passed on to the future generations. And, you know, always whenever you do some kind of a therapy, any kind of therapy for that matter, consent is very important. Consent is permit of that individual is very important. And when you're doing a germline therapy and you're bringing about changes in an individual who is about to be born, who is not born yet, you are doing things, you are manipulating things without the consent of that person is the first ethical issue. Now you would say that, how does it matter? It matters because please understand that, as I said several times, the genes that are inserted, they may not be inserted at a specific site. There could be a lot of non-specific insertion that takes place. And when non-specific insertion takes place, we need to understand it could disrupt a functional gene in that individual. क्योंकि अगर किसी फंक्शनल जीन के बीचों बीच मैंने ये घुसा दिया और उस फंक्शनल जीन को डिसरप्ट कर दिया, तो उस फंक्शनल जीन का तो अब बैंड बच गया ना. It will not be able to function effectively. So एक चीज से बचाने के चक्कर में मैंने उसको दूसरा जेनेटिक डिसऑर्डर दे दिया है. काफी सारे केसेस में जब ये नॉन स्पेसिफिक इंसर्शन डीएनए के ऐसे साइट्स पर होते हैं जो प्रोटोऑन्कोजीन बेस्ड होते हैं, then it could trigger cancer, could trigger tumor formation. Several of these insertional mutations could also lead to teratogenic consequences. And you know, it's something that is not ethical, because you are harming a new life, you are giving it some kind of disorder which it would have not suffered from if this therapy would have not been done. So preferably, you know, people have to be very careful when they do this to the germline cells. And also, it is strictly done only to an embryo which has chances of abnormalities. The gene therapy is going to be very crucial and has a hope of enhancing the quality of life of that particular fetus. Only under those circumstances, gene therapy is allowed. Because people use this technology for all wrong purposes. Okay, and when I say wrong purposes, let me tell you that there is a concept of designer baby. I don't know if we have discussed this earlier, we would have. Designer baby is a concept wherein a couple goes to an IVF clinic. They can normally conceive. They have the potential to normally conceive, but they don't want to do it, because they want specific characteristics in their baby to be seen. Like, मेरे बच्चे का हाइट इतना होना चाहिए, मेरे बच्चे के बाल का कलर ये होना चाहिए, मेरा बच्चा का आईक्यू ये होना चाहिए. ये सब कुछ जेनेटिकली मॉडिफाई किया जा सकता है. बट तुम कौन होते हो ये डिसाइड करने वाले कि वो बच्चे का फेट क्या होगा? तुम वो बच्चे को एथलीट बनाना चाहते हो, इसलिए उसकी टांगे लंबी होनी चाहिए. शायद उसको स्पोर्ट्स में इंटरेस्ट ही ना हो. Who are you? Is the basic question that we all should focus on, right? And this was indeed practiced. I am not fakification नहीं कर रहा हूं मैं इधर. ये seriously practice किया जा रहा था यूएस में, and ये ridiculous practice है, because this is not correct. You are no one to take a decision for someone else, right? So the designer babies concept was very, very much evident, and finally the US court brought a ban to it and said that you cannot use these therapeutic techniques until and unless there is some kind of abnormality in the embryo and gene therapy is required. Stop doing all this for nonsensical requirements and dreams of the couple, because this process has risks involved. So a normal baby unnecessarily would have to face consequences if something goes wrong. So that's unacceptable, right? So I will play a video about this. Just give me a minute. So this video that I'm going to show you is thoda sa bada, but we will try to see how much we can see it. It speaks about what potential genetic engineering has and how it's going to change life. Okay, so let's see what it exactly says.
1980s and were told that computers would soon take over everything from shopping to dating and the stock market. That billions of people would be connected via a kind of web, that you would own a handheld device orders of magnitude more powerful than supercomputers. It would seem absurd, but then all of it happened. Science fiction became our reality and we don't even think about it. We're at a similar point today with genetic engineering. So let's talk about it. Where it came from, what we're doing right now, and about a recent breakthrough that will change how we live and what we perceive as normal forever. Humans have been engineering life for thousands of years. Through selective breeding, we strengthened useful traits in plants and animals. We became very good at this, but never truly understood how it worked, until we discovered the code of life, deoxyribonucleic acid, DNA. A complex molecule that guides the growth, development, function, and reproduction of everything alive. Information is encoded in the structure of the molecule. 4 nucleotides are paired and make up a code that carries instructions. Change the instructions and you change the being carrying it. As soon as DNA was discovered, people tried to tinker with it. In the 1960s, scientists bombarded plants with radiation to cause random mutations in the genetic code. The idea was to get a useful plant variation by pure chance. Sometimes it actually worked too. In the 70s, scientists inserted DNA snippets into bacteria, plants and animals to study and modify them, for research, medicine, agriculture and for fun. The earliest genetically modified animal was born in 1974, making mice a standard tool for research, saving millions of lives. In the 80s, we got commercial. The first patent was given for a microbe engineered to absorb oil. Today, we produce many chemicals by means of engineered life, like life-saving clotting factors, growth hormones and insulin, all things we had to harvest from the organs of animals before that. The first food modified in the lab went on sale in 1994, the Flavor Saver tomato, a tomato given a much longer shelf life via an extra gene that suppressed the buildup of a rotting enzyme. But GM food and the controversy surrounding them deserve a video of their own. In the 1990s, there was also a brief foray into human engineering. To treat maternal infertility, babies were made that carried genetic information from 3 humans, making them the first humans ever to have 3 genetic parents. Today there are super-muscled pigs, fast-growing salmon, featherless chickens, and see-through frogs. On the fun side, we made things glow in the dark. Fluorescent zebrafish are available for as little as $10. All of this is already very impressive, but until recently, gene editing was extremely expensive, complicated, and took a long time to do. This has now changed with a revolutionary new technology now entering the stage: CRISPR. Overnight, the costs of engineering have shrunk by 99%. Instead of a year, it takes a few weeks to conduct experiments, and basically everybody with a lab can do it. It's hard to get across how big a technical revolution CRISPR is. It literally has the potential to change humanity forever. Why did this sudden revolution happen, and how does it work? Bacteria and viruses have been fighting since the dawn of life. So-called bacteriophages or phages hunt bacteria. In the ocean, phages kill 40% of them every single day. Phages do this by inserting their own genetic code into the bacteria and taking them over to use them as factories. The bacteria try to resist, but fail most of the time because their protection tools are too weak. But sometimes bacteria survive an attack. Only if they do so can they activate their most effective antivirus system. They save a part of the virus DNA in their own genetic code in a DNA archive called CRISPR. Here, it's stored safely until it's needed. When the virus attacks again, the bacterium quickly makes an RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. The protein now scans the bacterium's insides for signs of the virus invader by comparing every bit of DNA it finds to the sample from the archive. When it finds a 100% perfect match, it's activated and cuts out the virus DNA, making it useless, protecting the bacterium against the attack. What's special is that Cas9 is very precise, almost like a DNA surgeon. The revolution began when scientists figured out that the CRISPR system is programmable. You can just give it a copy of DNA you want to modify and put the system into a living cell. If the old techniques of genetic manipulation were like a map, CRISPR is like a GPS system. Aside from being precise, cheap and easy, CRISPR offers the ability to edit live cells, to switch genes on and off, and target and study particular DNA sequences. It also works for every type of cell, microorganisms, plants, animals or humans. But despite the revolution CRISPR is for science, it's still just a first-generation tool. More precise tools are already being created and used as we speak. In 2015, scientists used CRISPR to cut the HIV virus out of living cells from patients in the lab, proving that it was possible. Only about a year later, they carried out a larger-scale project with rats that had the HIV virus in basically all of their body cells. By simply injecting CRISPR into the rats' tails, they were able to remove more than 50% of the virus from cells all over the body. In a few decades, a CRISPR therapy might cure HIV. And other retroviruses, viruses that hide inside human DNA like herpes, could be eradicated this way. CRISPR could also defeat one of our worst enemies, cancer. Cancer occurs when cells refuse to die and keep multiplying while concealing themselves from the immune system. CRISPR gives us the means to edit your immune cells and make them better cancer hunters. Getting rid of cancer might eventually mean getting just a couple of injections of a few thousand of your own cells that have been engineered in the lab to heal you for good. The first clinical trial for a CRISPR cancer treatment on human patients was approved in early 2016 in the US. Not even a month later, Chinese scientists announced that they would treat lung cancer patients with immune cells modified with CRISPR in August 2016. Things are picking up pace quickly. And then there are genetic diseases. There are thousands of them, and they range from mildly annoying to deadly or entail decades of suffering. With a powerful tool like CRISPR, we may be able to end this. Over 3,000 genetic diseases are caused by a single incorrect letter in your DNA. We are already building a modified version of Cas9 that is made to change just a single letter, fixing the disease in the cell. In a decade or two, we could possibly cure thousands of diseases forever. But all of these medical applications have one thing in common. They are limited to the individual and die with them, except if you use them on reproductive cells or very early embryos. But CRISPR can and probably will be used for much more. The creation of modified humans, designer babies, and will mean gradual but irreversible changes to the human gene pool. The means to edit the genome of a human embryo already exist, though the technology is still in its early stages, but it has already been attempted twice. In 2015 and 2016, Chinese scientists experimented with human embryos and were partially successful on their second attempt. They showed the enormous challenges we still face in gene editing embryos, but also that scientists are working on solving them. This is like the computer in the 70s. There will be better computers. Regardless of your personal take on genetic engineering, it will affect you. Modified humans could alter the genome of our entire species because their engineered traits will be passed on to their children and could spread over generations, slowly modifying the whole gene pool of humanity. It will start slowly. The first design of babies will not be overly designed. It's most likely that they will be created to eliminate a deadly genetic disease running in a family. As the technology progresses and gets more refined, more and more people may argue that not using genetic modification is unethical because it condemns children to preventable suffering and death and denies them the cure. But as soon as the first engineered kid is born, a door is opened that can't be closed anymore. Early on, vanity traits will mostly be left alone. But as genetic modification becomes more accepted and our knowledge of our genetic code enhances, the temptation will grow. If you make your offspring immune to Alzheimer, why not also give them an enhanced metabolism? Why not throw in perfect eyesight? How about height or muscular structure? Full hair? How about giving your child the gift of extraordinary intelligence? Huge changes are made as a result of the personal decisions of millions of individuals that accumulate. This is a slippery slope. Modified humans could become the new standard. But as engineering becomes more normal and our knowledge improves, we could solve the single biggest mortality risk factor: aging. 2/3 of the 150,000 people who die today will die of age-related causes. Currently, we think aging is caused by the accumulation of damage to our cells, like DNA breaks and the systems responsible for fixing those wearing off over time. But there are also genes that directly affect aging. A combination of genetic engineering and other therapy could stop or slow down aging, maybe even reverse it. We know from nature that there are animals immune to aging. Maybe we could even borrow a few genes for ourselves. Some scientists even think biological aging could be something that eventually just stops being a thing. We would still die at some point, but instead of doing so in hospitals at age 90, we might be able to spend a few thousand years with our loved ones. Research into this is in its infancy, and many scientists are rightly skeptical about the end of aging. The challenges are enormous, and maybe it is unachievable. But it is conceivable that people alive today might be the first to profit from effective anti-aging therapy. All we might need is for someone to convince a smart billionaire to make it their next problem to solve. On a bigger scale, we certainly could solve many problems by having a modified population. Engineered humans might be better equipped to cope with high-energy food, eliminating many diseases of civilization like obesity. In possession of a modified immune system, with a library of potential threats, we might become immune to most diseases that haunt us today. Even further into the future, we could engineer humans to be equipped for extended space travel and to cope with different conditions on other planets, which would be extremely helpful in keeping us alive in our hostile universe. Still, a few major challenges await us, some technological, some ethical. Many of you watching will feel uncomfortable and fear that we will create a world in which we will reject non-perfect humans and pre-select features and qualities based on our idea of what's healthy. The thing is, we are already living in this world. Tests for dozens of genetic diseases or complications have become standard for pregnant women in much of the world. Often the mere suspicion of a genetic defect can lead to the end of a pregnancy. Take Down syndrome, for example, one of the most common genetic defects. In Europe, about 92% of all pregnancies where it's detected are terminated. The decision to terminate a pregnancy is incredibly personal, but it's important to acknowledge the reality that we are pre-selecting humans based on medical conditions. There is also no use in pretending this will change, so we have to act carefully and respectfully as we advance the technology and can make more and more selections. But none of this will happen soon. as powerful as CRISPR is, and it is, it's not infallible yet. Wrong edits still happen, as well as unknown errors that could occur anywhere in the DNA and might go unnoticed. The gene edit might achieve the desired result, disabling a disease, but also might accidentally trigger unwanted changes. We just don't know enough yet about the complex interplay of our genes to avoid unpredictable consequences. Working on accuracy and monitoring methods is a major concern as the first human trials begin. And since we've discussed a possible positive future, there are darker visions too. Imagine what a state like North Korea could do if they embraced genetic engineering. Could a state cement its rule forever by forcing gene editing on their subjects? What would stop a totalitarian regime from engineering an army of modified super soldiers? It is doable in theory. Scenarios like this one are far, far off into the future, if they ever become possible at all. But the basic proof of concept for genetic engineering like this already exists today. The technology really is that powerful. While this might be a tempting reason to ban genetic editing and related research, that would certainly be a mistake. Banning human genetic engineering would only lead to the science wandering off to a place with jurisdiction and rules that we're uncomfortable with. Only by participating can we make sure that further research is guided by caution, reason, oversight, and transparency.
Okay, so I hope that elaborate video gave you an idea of what all could happen. Though I would not support defaming any specific country as such, as it was shown in the video. But yes, we know that it could be used as a bioweapon too, right? So that is going to be really very difficult. Sir, I heard about talents, which is similar to CRISPR. People talk about CRISPR over talents. Yes. So we will surely speak about this when we talk about CRISPR-Cas editing. And that time we will compare both CRISPR as well as talents, Chinmay. So you will have to wait for 2 more lectures and then we will be coming to this aspect. Okay, Chinmay? Yeah, great. Okay, then I will stop at this point. We will meet tomorrow and we will continue with our discussion on gene therapy. Thank you so much. If you have queries, please feel free to put a message on WhatsApp. I am not taking it up right now because I know that you have a lecture scheduled after this. Thank you so much.